All right, I'd like to thank you guys for joining us today. Hopefully you all can hear me. First of all, I'd like to thank all of our speakers for speaking today. We really appreciate them taking the time and effort to put together a presentation for everyone. I'd like to thank Carrie Arnold for her patience throughout this whole process, actually, of getting this meeting together. This originally was supposed to be an in-person meeting, as I'm sure you all know. We've had to go to a virtual format, and it took us a little bit to find a date and everything, and she really helped us to get the word out to folks. I really appreciate her help. We have a new person helping us on the Extension Committee at UC Davis, and her name is Caroline Furman, and I'd like to thank her for her help with the webinar today and many things going forward. I'd also like to thank Anita Oberholster and also our department IT team who've helped us set up this webinar today. So without further ado, we'll get started. Our first speaker today is Megan Bartlett, and she is an assistant professor in the Department of Viticulture and Enology here at UC Davis, and she's going to be speaking about evaluating traits to improve rootstock through drought tolerance. Take it away, Megan.
Thanks so much. Thanks, Karen, Caroline, and Anita for planning this, and thank you all for coming. So can everyone see my screen? Okay, I'm going to assume so. Okay, so thank you so much. Today, as Karen said, I'm going to be talking about some work on identifying traits that we can use to improve drought tolerance in rootstocks. And so California is naturally an ecosystem that goes through periods of drought and especially strong heat stress. And in the future, as population centers continue to grow in California, competing with those sources of water use for irrigation is going to increasingly limit the amount of water that we have for our uses in agriculture. So improving the ability of crops to handle drought stress is going to be an increasingly important priority in the future. So in order to do that, we need to identify traits that can improve the performance of grapevines under water stress. And so one of the major tools we've developed to do that is through rootstocks. So the goal of our study was to evaluate traits in rootstocks that seem to be especially effective at conferring drought tolerance. And so what we want to do is look at variation in the rootstocks that are currently commercially important in California to get an understanding of what traits are important to maintaining the photosynthesis and the growth of the scion under water-stressed conditions. And so in this study, we really zoomed in on very fine-scale cellular traits. I'm going to talk more about those in a minute. But part of the reason behind this was inspired by work by Andy Walker, who's been extremely successful in developing disease-resistant cultivars. And one of the ways that he's been able to do this is to identify genes that are indicative of disease resistance. And we don't really have an ability to do that right now for drought tolerance. So that really slows down our ability to screen through large amounts of material for improved drought tolerance. And one of the reasons that we haven't been able to do that with drought tolerance is that a lot of different traits are implicated in drought tolerance. And a lot of these traits are involving many different aspects of the plant. They're involving changes in plant anatomy, structure, the molecular signaling, the chemistry of the plant. And so these traits often involve dozens or even hundreds of genes. So it's really difficult to identify a very small parsimonious set of genes that you can target in breeding efforts. So our goal in this study was to really zoom in on some very fine scale traits with the hopes that down the road, if these traits prove to be effective, they would be related to a relatively small set of genes that we could screen for effectively in new plant material. And so the traits that we were focusing on are traits that describe the cellular drought tolerance in the roots. And so we chose these traits because they're very important to the overall sensitivity of the rootstock to drought tolerance. So the roots are a major bottleneck for water transport through the vine, especially under water stress, where the below ground part of the plant can account for up to 90% of the total resistance to water movement through the whole vine. And this percentage becomes increasingly large as the plant becomes water stressed. So the roots are a major bottleneck for water movement during drought. And so most of that resistance to water movement up to the canopy is coming from this narrow band of living cells inside the root. And so these are these cells that are in between the outside of the root that's connected to the soil, and then the water transport tissue, or the xylem, that brings the water up to the rest of the plant. So in order to get to that water transport tissue, the water has to cross this network of cells in between the soil and the xylem. And so this is a major source of resistance to water movement. And as those cells suffer from water stress, they become even more resistant to water movement. So in this study, we wanted to really zoom in on the properties of those cells and get an idea of what would make those cells more drought tolerant. And so what's actually happening in these cells under water stress? So you can see that from work that's been done in the department by Andrew McElrone's group. And so these are some examples of what's happening. So the image in the center of the slide is showing changes in the cross-section of the root under a well-watered treatment, which is the control on the left, and then under a water stress of negative 1.5 megapascals on the right. And so what you can see is that the root starts experiencing cell death and cell collapse fairly early on in water stress. And so this causes a shrinking in the root that kind of separates the root away from the soil. So you can see that the root is kind of shriveling away from the soil in this image on the right. And so this is also having effects on water movement through the inside of the root, where as the water moves through the root, each one of these cells kind of provides an alternate pathway to water movement. But under water stress, as these cells collapse and die, the root starts losing pathways for water movement. So it encounters more and more resistance to the movement of water through the root. So under water stress, this cell collapse and cell death is causing two things to happen. It's making it more difficult for water to move from the soil to the surface of the root because it's shriveling up the root. And then even if water does get to the surface of the root, it's making it a lot harder for water to move from that surface to the tissue that's going to send it up to the rest of the plant. And so our goal in this study was to identify traits that are going to characterize these responses in the root cells, and then to measure and see whether these are related to the performance of the whole vine under water stress. And so we characterized the drought tolerance of these cells in two ways. So in one way, we were thinking about the volume of the cells. We were thinking about how much the root is physically shrinking as it loses water. And the other way we're thinking about this is the water pressure inside of the root cells. And so we're thinking about this in terms of turgor pressure. So what turgor pressure is it's the force of water molecules hitting the inside of cell walls. So you can think of this as the force of water kind of holding up the sides of a plastic pool. That force is turgor pressure. And so as the cells lose water, they lose this turgor pressure and the walls start to collapse in on these cells. And so ultimately, when you've really lost a lot of water, you lose all of your turgor and your entire cell collapses. And so we wanted to measure what's the sensitivity of the volume of the root and the turgor of the root to water stress. And so we did that in two ways. So we measured two traits that I'm going to focus on in this talk. And so one is the capacitance, which is the rate of the shrinkage in the root as the water potential inside the root becomes more negative. So you can think of this as kind of how much shriveling is happening for a given change in the water potential around the root. So in this case, a larger capacitance means that the root shrinks more. So we would expect that a more drought tolerant root actually has a lower capacitance. The other thing that we measured was the turgor loss point, and that's measuring the water potential at which you lose this turgor pressure and the cells collapse. And so in this case, we would expect a more drought tolerant rootstock to have a more negative turgor loss point, with the idea that this maintains the turgor under conditions that are drier and have a more negative water potential. And so the objective of our study was to measure these traits across a range of commercially important rootstocks, first to see if these even vary across rootstocks. Is there genetic diversity that we can work with if we're going to breed new rootstocks in the future? The second goal was to see whether or not these traits are always the same, or whether rootstocks can actually change these traits when they're subjected to drought stress to improve the drought tolerance of the roots. Because plants are continually shedding roots and growing new roots, we wanted to get an understanding of whether the rootstocks can be responsive to these conditions at all in changing these traits. And then the third goal was to see, as our hypothesis actually supported, that a more negative turgor loss point and a lower capacitance is actually making these rootstocks more drought tolerant, or these vines maintaining more water uptake from the soil and greater photosynthesis under drought. And so we did this as a series of pot experiments in order to really control the environment on these vines. But we also wanted to get an idea of, are these traits telling us about drought tolerance in field conditions? When we look at the rootstocks that have been characterized as drought tolerant or drought sensitive in field conditions, do we actually see that these traits can predict which one is in which category? And so what we did is we selected a series of 8 different rootstocks, the 4 of which are considered to be highly drought tolerant under field conditions, 4 of which are considered to be drought sensitive. So the tolerant ones include 110R, 140 rupestris, Ramsey, 1103 Paulson, and then the drought sensitive ones include 420A, 10114, Riparia, and 5C. And so we grafted all of these onto the same scion, which is Chardonnay. And then we grew these plants as potted vines in a greenhouse. So the reason we chose to do this as a pot experiment is we wanted to really standardize the drought treatment that we gave to each vine. So we wanted to eliminate the effect of rooting depth and being able to kind of escape a drought stress by growing deeply into the soil that some rootstocks have especially. So the way we impose these treatments is we weighed and watered these pots several times a week, and we maintained the soil water content at around 100% for the well-watered treatment, which is shown in blue here. This is an example of the relative water content of the pots. And then we allowed the drought treatment pots to dry out to a 40% relative water content, and we maintained them there over the course of the experiment. And so what we did is, for each combination of rootstock and watering treatment, we would destructively harvest plants to measure the volumetric shrinkage and the turgor loss point in the roots. And then for the remaining vines, we would measure gas exchange and water potential and the evapotranspiration of the whole plant over the course of the experiment. So this is an example of what that data looks like, where we're measuring the stomatal conductance at all these different points for a well-watered and a water-stressed plant. And then this is an example of measuring the water potential. And so what did we actually find? So first, we found that these traits actually vary significantly among commercially important rootstocks. So that's important because it says that there's a lot of genetic diversity to work with if we were to try to breed new rootstocks with different values of these traits to use under future conditions. And so this is turgor loss point on the left. This is capacitance on the right. And the blue bars are indicating a well-watered treatment. The gray bars are indicating the water-stressed treatment. So you can see that the turgor loss point is varying a lot from these very sensitive values up around 420A. That's something the root cells are going to collapse at a water potential that's still pretty close to 0. 110R is something that's going to keep turgor in the root cells and kind of keep those cell walls propped up until much more negative water potentials or much drier conditions. You can see the same pattern looking at capacitance, where 420A is going to experience a lot of shrivel in these roots. They're going to collapse very quickly under water stress. And then varieties like 110R are experiencing a lot less of this shrinkage. They're holding on to their volume under stress. So the other interesting thing that we found was that the rootstocks actually have a very large capacity to change these traits under water stress and to make themselves more drought tolerant, where you can see that there's a big shift in these traits to becoming more drought tolerant. The turgor loss points are more negative. The shrinkage is a lot smaller under these water stressed conditions shown in these gray bars. And so then the next thing we wanted to know was, does this actually improve your stomatal conductance and your photosynthesis during drought? So each of these points is kind of the average value for stomatal conductance for each rootstock over the whole experimental period. Blue points are well watered, black points are droughted. And so we didn't see a relationship between the stomatal conductance and the turgor loss point, but we did see a relationship with capacitance, or the root shrinkage, where we were seeing that the rootstocks that underwent the most shrinkage, that had the most root shrivel, are the ones that under dry conditions lost stomatal conductance the fastest. So they were forced to close their stomata and stop bringing up water from the soil. And this didn't really have much an effect on under well-watered conditions, as you might expect. We saw the same thing when we were looking at photosynthesis, where the turgor loss point wasn't really that related to the photosynthesis, but the roots that experienced the most shriveling underwater stress are the ones that did the least photosynthesis during drought. And there wasn't that much of an effect of the well-watered conditions. And then finally, we wanted to know, does this correspond to what we call water stressed or drought tolerant or drought sensitive under field conditions? And so this was actually really interesting, where when we looked at the trait values for the well-watered varieties, this is drought sensitive on the left, drought tolerant on the right, we actually found the pattern we would expect, where the rootstocks like 1103P have a more negative turgor loss point under well-watered conditions. But when we looked at the water stressed values, we actually saw that there wasn't really a relationship between the traits and how well the rootstock performs under droughty conditions. And it actually, the rootstocks that were the most drought tolerant in field conditions actually had worse drought tolerance traits under water stressed conditions. They're actually shifting to make their turgor loss points were not as effective at becoming more negative under water stress. So what did that mean? So we thought about it this way, this kind of discrepancy between the two experiments, where in the pot experiment, we've kind of subjected the whole rootstock to the same amount of water stress. And so if you are a root that is doing well at holding on to your volume under water stressed conditions, you're going to be kind of continuing your water uptake. You're going to be performing well in this entire soil profile because it's a very narrow soil profile inside of a pot. So this means that you're water stressed kind of evenly everywhere. And so being able to withstand water stress is beneficial to you throughout your soil profile. But if you think about in a natural soil profile where grapevines are very deeply rooted, there's actually a lot of heterogeneity in that environment, where there's roots that are dry up at the top, and then there's roots that are still wet down at the bottom where the soil is deeper and wetter. And so what we found in that case is that the most drought tolerant rootstocks in field conditions, what they're doing is they're really holding on to this volume under very mild water stress. They're performing very well in what we would expect to be these deeper, wetter soil layers. And they're allowing these roots to collapse and shrink in these dry soil layers. So they're not maintaining these roots up at the surface. Really, they benefit the most from having this function this function down in the deeper soil layers. So this was kind of interesting to us. Like, why wouldn't you want to just have your roots performing well everywhere in your soil? Why would you want to kill your roots off up at the surface of the soil where it's drier? And so we thought about it this way, where the roots actually have the ability to kind of move water from wet soil zones to dry. And that actually allows the roots in the drier soil to stay alive for longer. But the problem is that keeping these roots alive actually uses up a lot of carbon. So the respiration of the roots can actually account for up to 70% of the total photosynthesis of the grapevine. So it's very expensive to keep these roots alive. So the most drought tolerant rootstocks actually seem to have this strategy where they deliberately lose function and senesce these roots in the drier layers of the soil, potentially in order to concentrate carbon and root growth into the wet soil where it's going to be the most productive. And so that kind of pans out with what Andy Walker has seen in his work, where this really drought tolerant rootstock 110R, as soon as you rewater it after a drought, it expands a ton of area of root growth into the wet soil compared to something like Riparian. And so this strategy of killing off these dry roots and not paying the carbon to keep them alive is potentially a strategy that improves the drought tolerance of the whole rootstock. So for some future directions for this work, we're interested in whether this strategy of dumping roots in dry soil is always going to be the best in all environmental conditions. As California continues to get drier, is this still going to be a beneficial strategy? And is this also a beneficial strategy under all soil types and all irrigation management systems? We tend to be watering this soil that's up at the top of the soil profile. So is there potentially a benefit to kind of holding on to these roots in dry soil under some irrigation or climate conditions? How can we improve on these trait values? Would it help us to make these roots that stay in the dry soil even more sensitive, so we speed up this turnover and this dumping of roots? Would it help us to kind of find a more intermediate strategy where they're holding on to some of the roots in the dry soil, but not all? That's work we're going to be looking into in the future. And then what are some other traits that we can target at the same time? We've been really focused on this very cellular level idea of drought tolerance, but how can we integrate this with other traits that describe maybe the whole rootstock or the structure of the whole plant to improve the overall drought tolerance of the rootstock? So thank you all for listening. I'm really happy to take any questions at the end of the period. So thank you all very much.
All right. Up next, we have Elizabeth Forrestel, and she's an assistant professor in the Department of Viticulture and Enology, UC Davis. And I should mention that Megan, Beth, and the next speaker, Mason, all of them are new professors and have been hired in the last year or so. And in addition, if you guys do have questions and you would like us to ask them, there's a Q&A down the bottom of your screen. So if you want to write something in the Q&A, we can always try to ask those questions of the speakers. If we don't have time right when they finish, we can do it either during the break that I have sort of midway, or I've allowed some time at the end for questions as well. All right, without taking up too much more time, we have heat waves, vines, and wine impacts and mitigation strategies for warmer climates. Take it away.
Thank you, Karen and Caroline and Anita, for organizing this. And thanks so much, Carrie Arnold. I wish that I could see you in person and talk to you all in person. So today I'm going to talk about some work that we have ongoing in the Central Valley. Oh, am I muted? No, I'm good. Okay. Today we have some work, or today I'm going to talk about some work that we have ongoing in the Central Valley related to climate change and heat waves and impacts on vine physiology and berry chemistry. So a lot of work to date has, at least as far as in the ecology realm, and I'm coming from an evolutionary ecology background and also in the wine world, I would say, is really focused on these mean changes in temperature. And we know that there's this pretty rapid increase of warming in mean temperature, mean annual temperature. But what I'm most interested in is how, from a viticultural perspective, we can actually cope with extreme heat events, and that would be heat waves, droughts, and there's actually a more rapid increase of extreme temperatures. So nighttime temperatures, this is just here on the right-hand side is average summer T-min. You can see it's pretty rapidly increasing, and this is based on weather stations across California. So this isn't interpolated data, this is actual weather data. And on the left, you can see increases in T-max as well, daytime T-max. And this is for during the growing season. And if you actually made this go out to 2020, you'd see an even more rapid increase. And so what I am particularly interested in is understanding how, when we have these heat events, and the way that for the sake of this study, because it's in the Lodi area, the way we defined a heat wave or heat event is any 3-day period with a maximum temperature above 38 degrees Celsius or 100 degrees Fahrenheit. The way we determine that is based on regional historical weather. So we want to be able to relate what an abnormal event would be. So you'd create your distribution of average weather over the past 30 years, a climate normal, it's called, and then you'd say what is in the outer range, the upper 98th percentile in terms of Tmax. And so in the context of Lodi, that's above about 100. Obviously in areas in Stanislaus County, that's even higher, down near Modesto and other regions. But for the context of this, I'd say we also know too that above this temperature, grapevines are starting to suffer from other work. So in the context of all of California, you can see that relative to the 1981-2010 climate normal, this is the increase in the number of 3-day heat waves as per our definition of greater than 100 degrees Celsius that we'll see. You can see there's a really dramatic increase throughout the Central Valley. And similarly, one thing that's really critical is to not only think about warming in isolation, but also thinking about it in terms of water availability and water demand. And so on the right here, you can see how much of an increase there'll be relative to current time period to the end of the century in terms of potential evapotranspiration. And so we're going to be seeing this really intense increase of atmospheric water demand and increase in vapor pressure deficit, which will have a huge influence on plant function as well. And it means we'll be required to take more water to be able to cope with these heat waves in the Central Valley. And additionally, we know that there's reduced snowpack. So we have access to less water during critical periods. And even if our precipitation doesn't change, as far as mean annual precipitation, the timing of precipitation is critical to have these storage reserves for water. So there's a lot of future impacts and interaction between these that are going to be at play that are critically important. So when we think about lodi, specifically, which is where this study occurred, again, this is just number of days above 38 degrees Celsius annually, so it's not a number of heat waves, but it is about 3 to 4 heat waves, if you look at it that way. And you can see this is current 2050 and then 2100. And you can see this pretty dramatic increase where in some areas we have 0 to 5 days, at least in the, I should say current was 2090 to 2010 or 2080 to 2010. So we know we're out of that range now, where we're really seeing this rapid increase in number of days above 100. So we really do need to have on short time scales coping mechanisms. And so for a huge portion of my research program is coming up with these adaptive strategies for coping with climate change from both physiological and evolutionary perspectives. And I think about it in terms of both short-term adaptive measures, and this is what I'll be presenting to you today, is yes, we can talk about thinking about new varietals and what we would plant next and whether we need to develop new rootstocks and more heat-tolerant scions, but we also need to ask in the next 5 years, how are we going to cope with these current weather extremes? And so this is what I'm going to talk about today, is this mitigation of heat wave effects. I'm going to talk about irrigation manipulation, which I think is, other than shading, canopy manipulation is one of the primary means in the Central Valley. I realize there are other regions where you have other tools available, but it seems unlikely that nets or perhaps maybe, but at larger spatial scales, shading and misting is a little less practical. And I'm not going to talk about the cultivar-specific thermal tolerances today, but I'm also looking at that in the context of an experimental manipulation of heat waves in Davis and in possibly Napa. And then also other work that's ongoing, and I know I've contacted some of you who are here today about this, is this project to advance the Winkler Index and have a better measure of varietal suitability for different varieties based on wine quality and phenolics, starting in Napa with Cabernet and then moving to other regions, and then also integrating some AI perspectives with Mason Earles as well. And part of that too, we have common gardens that we're starting that are actually replicated and randomized in different regions. And so one part of this project is understanding what growers currently do as far as irrigation decision-making in the context of warming climates and heat waves. And so this is in collaboration with the USDA California Climate Hub and Lauren Parker, a postdoc there. And so I ask, we're sort of extended this survey to end later in August, and it's primarily focused in California. But it would be really great to get more from the Central Valley in terms of distribution across California. It only takes 10 minutes. And I'm going to show you briefly some of the preliminary data from this. So far, this is just from about 44 respondents. But I think it's interesting as far as what we should target in thinking about how to adapt to future conditions. And so this is on the upper left-hand corner. This just shows you the distribution of varieties that people are growing, people who responded. And it's largely, unsurprisingly, it's dominated by Cabernet and then Chardonnay. And actually I was surprised, Petit Syrah as well. And so it makes me feel like it's right to be focusing on Cabernet initially, especially for short-term adaptation strategies. And that's where the greatest value is as well. And then also too, you can see as far as growers, a large number of growers that responded had less than 50 acres. And then we also had this larger group where it was 500 more. So it was this bimodal distribution in terms of holdings, which is not surprising. That's how it generally looks for viticulture in California anyway. But what I thought was the most interesting that I wanted to share is when we asked what we thought the biggest issues, climate or weather related issues are for vineyard management, and this is across California, is what came out, and blue is not a problem all the way to purple, which is a devastating problem. So it was giving you 5 categories. And what came out as being the most relevant, we're thinking about drought and warmer daytime temperatures. So, and that doesn't say, obviously disease and smoke and wildfire and a few other things were important as well, but these came out to be the most relevant. And so I think that really speaks to the need for having strategies and greater knowledge of how we cope with these episodes of higher VPD, greater water demand, and heat waves. And so I'm going to share some of the results from our first year of trial. This is work in collaboration with Ian J. Gallo, with Nick DeCuzlia and Luis Sanchez, who've been wonderful about providing this variable rate drip irrigation site in Borden Hills. And I should say, I have to give Martina Galliano, a master student of mine, who has been managing this project and done a phenomenal job. And she just submitted her thesis, and we're working on a manuscript from this. So she has done an amazing job on this project, and I've been very lucky to have her. So this vineyard that we've been working in and are continuing to this season is all Cabernet. It's on 1103P, and critically, which I think is relevant to a lot of this audience, it's mechanized in terms of pruning, leafing, and harvest, and it's on high wire. And the way that this works is there's 30 by 30 meter pixels that can be differentially irrigated. This is a water bit system. And so we chose replicates across this vineyard, which is fairly heterogeneous in terms of topography and soils, and replicated a treatment of only during heat waves that we applied, which was keeping a baseline irrigation of 60% ET during the heat wave. And a heat wave, again, is defined as 3 days of temperatures above, maximum temperatures above 100 degrees Fahrenheit. And so we had the baseline, which will always be green in the results I show you, 2 times that, and then 3 times that amount of water. And that was applied the day before the heat wave, 1 to 2 days before the heat wave, and then through the heat wave. And so to show you what the season looked like, we had 2 heat wave events, and this is critically important because this will come up later, but the first one was pre-veraison. And so that occurred around July, end of July, mid to end of July, and that was 3 days. And then the second heat wave that we had was post-veraison, so August 15th, August 13th to 15th. And that was a little bit more of an extreme heat wave, but you'll see that we did apply these treatments. The irrigation, this is the irrigation going on throughout the season, and then we did double and triple during the heat wave events. And what we thought was interesting is that I'm not going to show you a ton of the physiological responses here. I'm going to dive into the yield and berry chemistry pretty quickly. But what was interesting is that we actually found that our most negative stem water potentials occurred prior to any of these treatments or heat waves. And so we didn't see a huge effect, a treatment effect at all during the heat wave in terms of additional irrigation applied, but during recovery we did see slightly less negative treatment, or less negative stem water potentials in the 3-time baseline treatment. But again, we didn't observe a ton of stress in the context of plant physiological responses as far as stem water potentials go, or leaf water potentials, and even pre-dawns too, which we did. But where we did see an effect in terms of thinking about net carbon assimilation and potential physiological effects on some of the source sink dynamics with the leaves and berries is we saw that so if we are thinking about actual temperature and function, this is, and the x-axis is the leaf temperature, and this is in degrees Celsius. And then on the y-axis is net photosynthesis, so carbon assimilation, and stomatal conductance, so whether the plants are keeping their stomates open and cooling. And you can see that in the baseline treatment, this yellow, that you did see an increase in temperature of the leaves and depressed photosynthesis, so depressed carbon assimilation. Similarly, you had a very significant reduction in stomatal conductance across all temperatures, and you reached warmer temperatures. And there was a slight difference between the 2x and 3x, but really the 3x didn't have as dramatic an effect as we thought it might. So it seems like we're hitting this limit of how much water you can apply to be helpful. And you'll see for some of the berry characteristics, you actually might have a negative response due to that. And so as far as yield, we did see an effect of yield, an effect of reducing water during heat waves on yield. We had a reduction in yield in terms of kilograms per vine and grapes per cluster. We also saw that in the baseline treatment where we had no additional irrigation, we reached higher bricks more rapidly. And this is in contrast to some results where with really high temperatures, you can see this delay in onset of maturity. I don't think it was warm enough for us to get that effect, but we did see that without that additional water, we had this increase in bricks even before we reached physiological maturity. So probably having an effect of the first heat wave. So this is just showing you the second heat wave here, but you see we reached about 25 bricks, which was our harvest time, or our target bricks for harvest, a week before, a little over a week before, which was pretty striking. Similarly, we saw an effect of the treatments on both pH, and this follows what you would expect: higher pH in the baseline, lower TA in the baseline, where there was more heat stress experienced. But what is interesting is you start seeing some of the effects of, not until the next one, but some of the effects of having too much water in terms of phenolics or secondary metabolites. And so this is total anthocyanins. We did dive into anthocyanins and look at the different types. I'm not going to present that here for the sake of time. But we did see that the baseline had the most significant reduction in anthocyanins. And interestingly, the treatment that performed the best in terms of amount of anthocyanins, in terms of milligrams per berry, was the 2 times baseline. And so we're starting to see that you have this negative effect of applying too much water. Again, not too surprising in terms of wanting to maintain deficit irrigation for some of these berry quality traits, fruit quality traits, but it is striking in the sense that all we did was manipulate for a total of 8 days in the season in terms of additional irrigation. And so it's this notion that we really can have a pretty dramatic effect in terms of decisions we make during heat waves. And again, we saw this decoupling of anthocyanins and sugars with too little or too much water. You see these trends here, and this is following work that's been done in Australia that found the same thing. And so lastly, I just wanted to mention too, which I think is really interesting, and we're investigating further from a molecular and transcriptomics perspective and doing more sampling this year and monitoring berry temperature, is that first heat wave had a lasting effect on total tannins through the season. So again, you see that this effect that precedes heat wave 2 of having a reduction in tannins, not surprisingly, synthesis is finished much earlier and initiated at flowering for tannin synthesis, as is different than anthocyanins. And so you see this reduction here where baseline is the lowest and 3x is the lowest. And I'm not going to, for the sake of time, I'm just going to mention quickly that we've only looked at wine 3 months post bottling, and we did see an effect, but it was primarily due to alcohol and then some parameters affecting potential fermentation dynamics, but not so much with the phenolics. But we, due to COVID, haven't been able to do our formal analysis of these wines. And so that'll be something that comes later, and then we're doing that this season as well. So as far as conclusions and what we're up to next, I think it was really critical to see that these different treatments, from a physiological perspective, we saw recovery. So you might not make much note of it, but we did see significant effects on berry composition, yield, and wine properties, and where negative results are observed from excess water. And we're trying to dig into why that may be a little bit more from a mechanistic perspective. And then also too, that we do see this higher evaporative cooling, but there's a limit. And so we want to be able to provide general guidelines in terms of how you do want to treat it based on the baseline, your soil moisture, and other properties, so that it could be used more generally. And with that, I would like to thank all of the collaborators and labs that we work with, our funding support, especially E&J Gallo for their cooperation and in-kind support, which has been really phenomenal. And then lastly, I just want to throw this up again to encourage you to take our survey. And thank you so much for all showing up. And hopefully I'll see you in person before too long, at least in the field. Maybe I'll see you in the field, in the vineyard.
Thank you, Beth. As far as the link goes, we may end up having to send that to the participants. I'm not sure that they can actually, the link goes anywhere in the actual chat itself. So we may just send that to the list of folks that signed up through A&R, because we'll have their emails and we can send it to them. All right, I don't see any questions. Let's see. Hold on a second. There might be something in the chat. All right, from Nick, you have a question about your data makes perfect sense based on current knowledge of berry development and gene transcription. Do you have a good feel for benefits from cooling effects versus physiological effects of water applied?
Do you want me to just answer it, or do you want to move forward and I can type the answer?
You are welcome to type the answer if you'd like. I think you have another one from Carmen as well.
Okay, I can type the answer so then people can just see it and you can move on with Mason.
Perfect. All right, we'll do that. If folks in the chat are good with that, then we'll move on and let Mason, you'll have to unshare your screen.
Oh, did I not do that?
Oh, no, you did. I'm sorry, you did. Sorry. Yep. All right. Well, we'll go on. I think it's just a question of changing. Let me see. Yep. There we go. All right. Next up is Mason Earles. He is another one of our new assistant professors in the Department of Viticulture and Enology, and he's also in the Department of Biological and Agricultural Engineering at UC Davis. And he is going to be presenting AI-enabled precision viticulture. Take it away.
Well, thanks a lot, and thank you to all of you for coming. I'm super excited to get your feedback and tell you a bit about what we've been up to in our lab in collaboration with some Beth and some of the other folks you've already heard of, and a number of you are online as well, such as Nick and others at E&J Gallo. So what I'm going to be talking about today is AI-enabled precision viticulture. And specifically, we're interested in this question about how we can use artificial intelligence to enable grape producers to more sustainably manage vineyards. And so that has to do with both questions on the yield and quality side, but then also related to that, how we can help enable less resource consumption and waste. And so that's just the big, broad kind of long-term mission of the lab, right? And so thinking about this, we're building on a number of big challenges that we found, myself and a number of others have been working to define as part of our efforts for AI-enabled agriculture more generally. And so those challenges, I think, apply really directly to the grape industry. And one of those is that production conditions in agriculture are highly variable, right? So in that we have all sorts of, in this case, grapes, we have many different cultivars, environmental conditions, management conditions. And each of these makes for a pretty variable data set that when you're trying to train AI algorithms, oftentimes what will happen is you'll have to capture this level of variability. So this is a big challenge. So this means either we need a lot of data or we need smarter algorithms that are tailored to agriculture specifically. So the second challenge that agriculture faces is that we often run into limited internet bandwidth, memory, and power supply conditions. So this is basically remote types of sensing conditions where we don't really have internet, in other words. So this actually makes a challenge for going to and from the cloud, right? So a lot needs to happen on the device locally and deliver results quickly. So the third challenge being that technological adoption actually, we think, requires ag producer confidence, as you might not be surprised to hear. And so what we mean by that is that in order to actually get adoption, you need data to train good models, and you need adoption to get data. And so there's kind of this chicken and egg interplay that has to get going to catalyze this sort of technological development. So these are the three big challenges that underlie a lot of the research that we're interested in. And specifically, we've been putting together kind of a roadmap for AI-enabled agriculture. And this, I'm not going to go into the great details on this, but basically it ranges all the way from how we build in 3D crop models and our previous knowledge of how crops grow and the state of the art for that into AI-enabled frameworks that then also take into account a lot of what we know is important, such as auxiliary data, including climate, weather, soil, environment, into these models that then can also bring in this type of sensing data as well, because all of this is important and it goes into the process of explaining things like yield and quality forecasting, which is what we're ultimately interested in, and robotic types of management. And so this is kind of the overall framework where try to situate the research in our lab and say, okay, what specifically are we focusing on in this bigger framework in the context of viticulture and grape production? So I'm going to talk more now specifically about a project we've been working on at this point for now about 8 months. And it's really from this AI-enabled sensing through the modeling, the machine learning modeling, up to the yield, ultimately forecasting right now, even just prediction. So this has been in collaboration with a number of other folks, including, again, E&J Gallo, who we did a lot of the data collection on initially, the sites at which we did data collection from initially. And so Alex Olinsky is a PhD student, and Pranav and Jung Hao are also working on this as well. So these are the 3 of the students that are working on this project. So I'm going to zoom into Borden Hills. Beth talked a bit about Borden Hills earlier. I'm going to talk some more about it. So we're going in. Here's kind of generally where we're at, and zooming in, this is what the vines roughly looked like when we were there. And so this is premised on the idea that they typically do mechanical grape harvest here. And so what, along with a lot of other people do, is if you're going to measure and map yield, these yield monitors typically use a scale, essentially, a load cell on the conveyor belt that's trying to measure the mass of the fruit as it passes along this conveyor belt. And so this is good because you can at least get an idea as you're harvesting roughly how much you're harvesting and how that's distributed throughout the field. But one of the challenges is that you're getting it at the time of harvest. There's a lot of calibration that's actually involved still in terms of, because this is rattling around while you're harvesting the whole time, it's hard to measure things, keep things calibrated while that's happening. And so what would be nice is we can actually do some more move towards forecasting earlier towards veraison, so that we're able to then have more of a runway to make management types of decisions on leading into harvest. So that's building on this. These harvesters are equipped with, again, GPS and load cell to measure the mass moving across the conveyor belt. And we use this ground truth data that Gallo's been generating to try to, what we're doing is trying to train a model to predict this yield. And so in this case, I'm going to skip over some of the details in the sake of time, but this is Alex. This is during our first month when we started. We had a very short amount of time left before harvest, so we went out and put together a really quick system to just grab some reasonably high quality video off of an ATV, record this, georeference it at about 1 meter resolution, and we would use that to then, again, try to say, well, can we ultimately predict the ground truth yield that was measured from the yield monitor? So this is what the video looks like, and again, you may be not seeing it at full quality, but we moved about 4 miles per hour, and from that we get a bunch of different images. And so the first thing we had to do was say, okay, well, what you end up with is a lot of data that's not usable. So a lot of unusable images. So these two, the left and the right, are examples of what you get quite a bit of, which you don't want to be using to predict yield, right? Because clearly at the end of the row, or when you have a big branch in front of the camera, that is not going to be accurate. So the first thing we did was train a model to just, it was a image quality model that kept only good, high quality images. So that was the first thing. So we just got rid of all these others. The next thing we did was had Alex went in and hand traced about 120 images worth of grapes to show the model, and this is called supervised learning, where the grapes are actually at. So after he did that, went in and we trained several different state-of-the-art, what are called neural network models. These are just machine learning models that have been exploding in popularity and success in the last few years. And from that, we ended up having this pipeline. And ultimately, it worked reasonably well in terms of its internal consistency. And I'll show you what I mean by that in a second. So what I mean is that we had 2 different models. Let's just say one predicts kind of, you can see these polygons or these shapes around where the grapes are at. The other one just predicts boxes. That's the only difference between the left and the right. This is a lot less computationally intensive. So what we end up with is basically predicting where all the grapes are at, or I shouldn't say all, we predict pretty well where the grapes are. And you say, okay, well, qualitatively, it seems fine. I can't see that well through your screen anyway. So we held out some percentage of those hand annotated images, about I think 25, that Alex had labeled. And we said, okay, we're not going to train the model on these, hold these out completely, and we'll see how well it does. And so there was pretty good internal consistency in terms of the ability of the model to predict what Alex drew as grapes. So the model did a good job predicting what it could see, right? We're not going to worry about that at the moment. The bounding box and the mass worked equally well. But the big challenge comes when you try to go from pixels to kilograms, right? So this is what the yield monitor measured, and this is what we're seeing as grapes and saying these pixels are grapes. So here, what you've got, I've taken out the numbers, but just you'll get the point very quickly. Yield monitors in orange, the image-based predictions in blue, and what you probably see is that they don't look the same, right? And so we went, okay. We looked at the data quite a bit and didn't see much of a correspondence, and we're starting to say, okay, well, what's going on? Who's wrong? So the first question was, well, we have to make sure that our ground truth is actually ground truth. So we went back and talked with Brent Sams, who works with Gallo as well, and said, okay, do you have any data where you guys have done some hand harvest or something that you like feels really good quality? He said, yeah, I've got some. So he checked that and just did a really quick correlation between the yield monitor and the hand harvest and said, okay, well, there's actually this, he didn't, no smoothing, nothing. There is a relationship. And if you've done some smoothing, and you could clean this up and tighten it up even more. So we're going, okay, well then we're wrong, right? Our model's wrong. The yield monitor is good ground truth. Okay. So that's promising at least. So then we started saying, well, what is it that's causing us to be wrong? And so we started going in and saying, well, let's look at the points where there are major discrepancies in the data, where we get like real high predictions in our images or low predictions on ground truth, or vice versa. So when we started doing this and we started just looking at the images and saying, well, what could be driving this? And this error in our model. And so what we started seeing is that you actually have things like, well, we know the distance to camera is an issue. So you can have grapes that are very close to the camera or further away, which we aren't accounting for yet. So that's an obvious one we've started dealing with already. There's occlusion and sparsity. So leaves can either cover up grapes that you can't see, grapes can cover up grapes and so on, or sparsity. So that would lead to undercounting. Sparsity, on the other hand, would be I can see through the canopy and I might be double counting grapes. Right. So if it's like a sparse, low leaf area index type of environment, you might be seeing grapes on the other side and double counting those. So you've got like multiple sources of error happening at the same time. Depth camera might help with that one. And then we've got averaging across sides. So the yield monitor is collecting multiple sides at one time. So we've got that being a potential challenge. And so how do we overcome these obstacles? And so some of what we're working on now, and spoiler alert, we haven't solved this problem yet. But I'm going to tell you about a bit of where we're at today. And that's using auxiliary data inputs and ag-specific AI models. And here, this is bringing in kind of these other types of information. And so we've been doing this in collaboration with Stavros Vizoukas at Davis and Beth Forrestel within the RMI vineyard. We have an international block where Beth's doing a lot of work already. So we piggybacked on that. And we're going in and kind of have built a version 2 of the sensing kit. And we have it roving around on a small robot once a week. And we've been doing this since the beginning, since about late March. So this is Junghao and Alex who've been doing this. I'm just going to play a short video. This is the robot roving through, collecting data. And what it's doing is actually, not only do we have data on phenology as well that Beth is already collecting. So we're going to be feeding phenology information, phenological state information into the model. We also have not only RGB, which is your red, green, blue standard imagery, we have IR and depth imagery, which should hopefully help with some of the issues we've talked about earlier, although I think time will tell. And additionally, we're taking another approach, which is just trying to get a lot more training data. This is the like Google, Facebook approach where you say, okay, well, let's just let the model learn from the data. But one of the big challenges is how do we get more data? So that's what we've been thinking more about as well. So the idea is to go ahead and take existing operations that are being done in vineyards and try to piggyback on times whenever you're already out in the field, driving a tractor through the field, right? So we chatted with different folks at a few different vineyards and found that probably the time to do that, where we would get the most data in the right window, would be during sulfur dust, periods of salt, when you're doing sulfur dusting activity, right? So that's about every 7 to 10 days. It's leading up to veraison. We might be able to get a good insight into the berry development in the earlier stages and identify where they're at. So knowing that, we still have a number of constraints from an engineering perspective that we have to think about. And so if we're going to mount on a sensor on existing tractor, we're saying, okay, every 7 to 10 days, typically this is happening at night. And the speed can range anywhere from 4 to 10 miles per hour after talking with a number of people who do this. So first we went out and we've been taking images at different positions from the tractor. And additionally, we've been collecting vibrational data and other types of motion blurring data, which are a major challenge when you're moving 5 to 10 miles per hour in a bumpy field at night, trying to take video. So we've got a kit this is the just a quick way of getting some data on the video footage and the vibrational data that we've been collecting as we're moving through the field at about, that's a 10 mile per hour run. So we've got data on that. And from there, we'll look at the vibrations and the motion. I did this also with an iPhone. Now we have it also corresponding with the imagery, so we can start engineering the right system, the right camera sensing system to get high quality images. And start building these multimodal types of models that we talked about earlier. And ultimately, the goal is to, again, convert pixels to kilograms. And to do this, we're focusing on not stopping at the point where we say, okay, these pixels are grapes, but we're trying to push forward new methods, which are kind of, I think pretty advanced as far as deep learning goes, to use regression models. So we're actually just trying to train the model to directly from these different types of images and weather data to predict a yield value, right? Not to stop at pixels and then try to sum them up and say, well, if you sum all the areas of the pixels that belong to grapes, this is what you end up with. We want to actually predict yield. So we feed in, the idea is to feed in the yield monitor data as the predicted, as what we're trying to predict, take in a bunch of different imagery and kind of local weather and climatic data and try to make predictions on yield. And if we can get enough data from being able to mount a number of these different sensors on tractors, I should mention we're trying to do it under about a $1,500 to $2,000 cost, then we think that that might enable a lot more data collection that we could use to take the bigger data approach. So these are sort of what we're hoping will be some of the solutions. And then in my last 20, 30 seconds, I just want to also mention one of the issues that we face in agriculture is not having enough. Labeling data is expensive. So the hand drawing that Alex did is really hard and time consuming. So one of the things we're doing in collaboration with Brian Bailey, a few others, is actually using 3D simulations to let the computer annotate the data in these 3D computer graphic generated worlds. So we generate 3D models and take a lot of infinite number of pictures, and we label data in a simulated environment. And then we go out and using the same data, we can make predictions in the real world, and we actually get pretty good predictions of where the grapes are, having never seen, the model never seen real life. It only was trained on simulations. And this is something people in autonomous vehicle companies, Uber, Lyft, and so on, have been doing for a while in terms of simulating self-driving cars. And so this is the idea of training and simulation can save a lot of money and time in terms of human effort. So that's something else we're interested in focusing on. So that's it. Again, this is kind of our general roadmap for AI-enabled agriculture, and I think there's a whole lot to explore here, and we're only just starting to scratch the surface. So I'm looking forward to all of your feedback, working with a lot of great collaborators at UC Davis and outside of the university, and please don't hesitate to reach out if you have any ideas or want us to try to collaborate together. We're really looking forward to that. Thank you.
Thank you, Mason. So one of the things I'm going to mention to folks, if you guys do have questions, the Q&A instead of the chat is a better place because it's easier for the panelists to answer those questions and to answer them for everyone to see the answers in the Q&A. So again, if you have any questions, feel free to ask them in the Q&A. And if you'd like it just to be answered here while we're here, please feel free to say that as well. All right. So I think we're just going to take maybe just a 1 or 2 minute quick break, and then we'll have Anita up. So we're just going to take about a 2-minute break, let people get a little water, a little bio break, whatever people need to do, and we'll be right back.
I'm going to try and keep an eye on the time, Karen, but I'm not actually 100% sure how long my presentation is. So just sort of give me a 2-minute warning.
Okay. You want me to text you?
Let me get my phone. I sometimes forget about my phone when I'm presenting.
All right. I'm thinking maybe people are now seated again. Let me see if, let's see. All right. So next up, we have Anita Oberholster. She's our Cooperative Extension Specialist in Enology in the Department of Viticulture and Enology at UC Davis. And today she is going to be speaking about cleaning and sanitation in the winery.
Thank you, Karen. Okay, let me quickly share my screen.
Mm-hmm.
And I'll quickly go in presenter mode.
We're all hoping.
Okay.
Yeah.
Okay, let me just remove everybody's faces so that I can actually see my own screen. Good afternoon. So I was asked to talk about cleaning and sanitation in the winery, and it is probably the most important step during winemaking, and it's probably also the least interesting part of winemaking. So I'm apologizing in advance, but I'm going to try and make it interesting. So cleaning and sanitizing is supposed to be a preventative process. It's not supposed to be a corrective one. So that is the first very important point. And yes, I'm putting minor time investment because we all know that during harvest, it feels like all we're doing is cleaning and sanitizing. But overall, if you think how long it takes to get your wine in that bottle, it's actually a minor time investment. And it's a really important time investment. To make sure you have cleaning and sanitization protocols that work, you need to establish written protocols to ensure product quality and worker safety. I'm not going to list them all. You can see the benefits as well as the consequences of not having a proper cleaning and sanitization protocol. So as wineries are now FDA regulated food processing facilities, this is for all aspects of winery processing. And cleaning and sanitizing in the winery is actually pretty difficult because we have equipment, large range of different pieces of equipment that can be very difficult to clean, made of a range of different materials that's more or less easy to clean. First step, what's cleaning? So cleaning is the actual process of physically removing organic and inorganic soils. Now, soils is just a fancy way, a scientific way to say dirt. Then you get your sanitizing. So sanitizing can be three different things. It can be disinfection, sanitization, and it can be sterilization. So what's the difference? The difference, if you look at the left, you'll see we focus in a winery on sanitization. Sanitization is a log 6 decrease in spoilage microbes. So it's not that there's no viable cells left. That would be a log 12 plus decrease, and that would be sterilization, something that you do in the operating table. It's not necessary in a winery. We just want to really limit the amount of microbes in the winery so it does not result in a problem down the line. Now, any chemical you use in the winery, you need to know the safety data sheet. You look at the safety data sheet to determine what personal protective equipment your people need to wear. That's also where you see what's the permissible exposure limit so that you know what needs to be the ventilation in the room that you're working with this equipment. Do they need masks, etc., etc. It's really important to follow these protocols, and it's based on the SDS sheet that you develop your standard operating procedures using these chemicals. Now we have the additional safety regulations due to COVID-19. I'm not going to talk too much about it, but the Wine Institute have a page called Health Safety Best Practices During COVID-19. It has some really useful information. Basically, it recommends that every winery should have a policy for employees to report illnesses, to refrain from work if they have any symptoms as specified by the CDC. And that list of symptoms grows every day. So you need to go have a look at what that is. Now, they have some guidelines there that's really interesting. And if you don't have protocols yet, I will go and read those guidelines. Ideas of what do you do when somebody tests positive? How you determine who had a close contact with somebody that tested positive? What do you do with that person? Should that person quarantine? Can they continue working? There's a lot of different options and guidelines there that's really, really useful so that you have something in place if you're faced with that situation in the middle of harvest. It's really important. The other thing is to remember, not all wineries are in the same county. Every county have their own guidelines, so make sure that you know the guidelines of your own county. Then let's move back to winery microbes, what actually is my field. So wineries have a high microbial load, especially during harvest. Now, luckily for us, master wine properties place selective pressure on microbes, meaning that we only have to worry about microbes that can cause spoilage, impacting wine quality. We don't have to worry in general about things that will make people sick if they drink your wine. This is because we work in a low pH environment and we have alcohol in our final product that causes a lot of pressure on microbes. The microbes we're looking at are also all heat labile, meaning that heat will denature them and inactivate them and kill them. So spoilage microorganisms requires a vector to travel through a winery, and these vectors are workers many times. This is why cleaning is very important. Your HVAC system, they need to have proper filters, improper sanitized equipment or tools, and obviously fruit flies. This is why we don't want fruit flies in a winery, and we have to clean up any spills. Now, 0.45 micron filtration does remove commercial yeast, but you have some wild yeast and bacteria that can go through there theoretically. So basically what I'm saying is, while filtration is a good idea if you think you have a bacterial problem, it shouldn't be your only remedy for a potential bacterial spoilage. This is just one of the things that you can do to improve your chances and remove most of the bacteria that's in your wine. So we're worried about yeast, malts, lactic and acetic acid bacteria. That's really our main sources of wine spoilage. And we need to worry about both planktonic. Planktonic is when it's suspended in the medium. Sessile or surface associated is what we call a biofilm. Okay. Now, biofilms are usually much more difficult to inactivate or remove. And in fact, you don't just want to inactivate. If you only inactivate the microbes in a biofilm, the architecture is still left, and very easily other microbes just go and stick on there and form an even stronger biofilm. So you really want to remove it totally. Now, usually, how do we assess microbial spoilage? Normally, it's through sensory. You figure out something is not exactly the way it should be. Then we can do plating and microscopy sometimes in the winery, depending on your setup, or you send a sample away for molecular methods like scorpions. If you look at plating or microscopy, you want to probably have an ID guide. It can be quite difficult to identify different microbes under a microscope, especially if you don't have a lot of experience. Now, I'm going to run through the different cleaning chemistries. I'm not going to read everything that's on these slides. I'll share my slides with everybody later, and you can look at this at your leisure. So our main caustics are our sodium or potassium-based caustics. Then you get your non-caustic or more milder base products. Those are often your sodium carbonates, potassium percarbonates, trisodium phosphates. Then you have your acid cleaners, they're phosphoric or nitric based. Sanitizing chemistries, we have a few more options. Chlorine based, mainly chlorine dioxide. It has not been associated with any TCA issues. Iodine-based compounds, you get your quaternary ammonium compounds, we many times just say quats. Sulfur dioxide. Now, sulfur dioxide is a sanitization agent we usually don't recommend because by the time it's really effective, you need to use pretty high concentrations, and there's some hazards to health at those concentrations. And peracetic acid has become very popular because it's a no-rinse sanitizer. It breaks down to innocuous compounds like acetic acid, oxygen, and water. Don't worry about the acetic acid. That is really negligible amounts. Ozone locked chlorine dioxide, you have to generate that on site. It has a pretty short half-life in water, so it works well, but if you generate your own ozone, make sure that you generate it very close to whatever you're treating. Heat and steam works really well. Thing to remember there is obviously it does add to your electricity bill. It does take a lot of electricity to run steam. Then UV, ultrasonic, there's a lot of other techniques that's not that commonly applied in the wine industry. There's been tests in laboratories to show that they work really well. Something to remember for something like UV. UV works really well, but if anything is in the shadow, it is not being sanitized. Just remember that. So UV works better for something like water that flows on your lab or a flat surface. So this is your standard basic 5-step cleaning and sanitization process. You usually have like a room temperature water rinse to get most of the dirt off, then a warm water rinse to get more of the salts that may be on the surface off. Remember not to use boiling hot water that can cook the dirt onto your surfaces and then makes it actually harder to remove. That's usually followed by a cleaning cycle and then another water rinse to remove the cleaner and to neutralize. Sometimes people will add citric acid to that water rinse. Obviously, that adds to the carbon oxygen demand of your wastewater. Just keep that in mind. It's not always necessary to acidify it. You can just use pH strips to see if it's neutralized, if your water rinse works well enough. Then you have your sanitizing cycle. And if you don't use a no-rinse sanitizing agent, then you need to do a final water rinse before putting wine or whatever in that piece of equipment again. So I've mentioned this a few times before, but repetition is key. You need written protocols, and then you need an employee checklist to make sure that those written protocols are actually being followed. The other thing is, don't just assume it works. The first time you develop cleaning and sanitization protocols in your winery, you need to monitor it. You need to check that it's actually working by using pH meters or pH strips to make sure that the pH of your solutions is actually what it's supposed to be. Using ATP and environmental swabbing to make sure, yes, I have actually cleaned all the dirt away. Just visually clean is not a good enough indicator. I'll show you data to prove this. Temperature tapes and thermometers. Is my hot water really hot? Okay, just to make sure. Is it really hot enough? And also, we recommend that you check this definitely just before harvest, somewhere between perhaps halfway through harvest and at the end or something, just to make sure your processes are actually working. So you need a hazard analysis and critical control point plan. It's basically what it is. It's a flow diagram of your winemaking processes. And then you identify critical control points. And critical control points are many times like your filtration station, your bottling line, your fermentation tank, and then for that critical control point, you will have your SOPs and your checklist that your employees need to check off. So we did this small study where we looked at the 7 main microbes in wineries that we need to worry about. We did this because we thought, okay, we're going to screen cleaners and sanitizing agents and see if there's some green alternatives to improve the quality of your wastewater. The other thing is also to see, can we use less of it so that we increase also the quality of your wastewater by having less chemicals in it? And can we perhaps minimize contact time to decrease the amount of water you use and decrease the amount of wastewater you're generating? So for 3 years, we look at Saccharomyces, Zygosaccharomyces, and Brettanomyces. For bacteria, we looked at Lactobacillus, Acidobacter, Pediococcus, and Unococcus. So first we did a planktonic cell screening. That's in the medium, not a biofilm, in the medium. And we used 96 well plates. We had 7 microbes and we had 20 different treatments. 9 cleansers, 10 sanitizers, and then our water was all blank. Basically what we did is we put the microbes in, we challenged them with the chemistries, we played it out every 5 minutes to see when the cells were not viable anymore. Now, if you think about this, this is a total unfair test to a cleaner. This is not what a cleanser is supposed to be doing. This is a fair test for a sanitizer. So this is something we did keep in mind. Next up, we tested biofilms. We formed biofilms by incubating the microbes in 50% grape juice for 10 days at 30 degrees. And then we challenged these biofilms with the different cleansers and sanitizers. And we stained the biofilm using crystal violet staining to see if the biofilm was still there after cleaning or sanitizing. Now, obviously, if you think about it, this is a fair test to the cleanser, but an unfair test to the sanitizer because the sanitizer needs cleaning first. But this was to screen because at this stage we had 20 treatments and we can't take 20 treatments to the winery. We just wanted to cut it down. The next step, we wanted to go something closer to our winery surface. So at this stage, this was plastic. So we went to stainless steel coupons and we grew the biofilms on there. Now the 3 years could form a biofilm on its own, but we couldn't get the individual bacteria to form biofilms. We had to mix them at the end. So we had the 3 years, one bacterial mix for biofilms. And what we did is we incubated it, formed our biofilms, took it off, rinsed it with water. It was visibly clean even before we started the cleaning process. Very important to remember this. And we basically shaped it in a Petri box, a pipette box, and then tested it with ATP bioluminescence swabbing as well with plate counts, plating it. And this is what we saw. Now, once again, this is biofilm, so this was an unfair test to the sanitizers. And if you look at the results here, at the right, all the high bars, that's all the sanitizers, okay? So this just affirms, even if something is visibly clean, you can't skip the cleaning step. You have to clean. Now let's focus, zoom in to the cleaning step, okay? So you can see we went down from 9 cleansers down to 5, or actually 6. Most of them, 4 of them, were caustic-based cleansers, and the mix was a proprietary blend of percarbonates and hydrogen peroxide and other things, wetting agents and surfactants. Basically what we did, we did both 5 and 10-minute contact time. Now the green dotted and the red dotted line, the 10 is the theoretical pass, the 30 is the theoretical no pass. This is what just the guidelines that they give. What you really need to do is figure out what is a pass and what's your range, your pass and no pass range in your winery. By taking your cleaning and sanitization protocols, applying it to the best of your ability, swabbing everything, plating in, looking what's in there and decide, okay, this is acceptable, and coming up with your own margins. That's what you need to do. Because you'll see, basically even the best one sort of failed for some microorganisms, okay? Then we did this test where we looked at the minimum inhibitory and minimum biocidal concentration. We did this for planktonic as well as biofilm in 96 well plates. Now, basically, what you're doing is you're doing a serial dilution and seeing at what concentrations were these cleansers and sanitizers still effective in inhibiting or killing the microbes. Now, this is a standard test that they do in the food industry, but it has a 24-hour contact time. So, and nobody has 24-hour contact time in the winery. I do realize that. We wanted to see what we're going to find. Interestingly, I would say the manufacturer's recommended concentrations were really generally the best. We put them on a very stringent test. So basically those where the biofilm was 100% killed and removed, those were the concentrations I looked like at, and because it was a 24-hour contact time, I wanted to at least be able to double that concentration to be sure, and then we were right back where the manufacturers, concentrations the manufacturers recommend. The next step was to take our top cleaning and sanitation protocols to something that's more winery dirt-like. So we took our stainless steel coupons, we put them in a custom-made device that fitted into the cap of our research fermenters. Okay. And we did tetraplicate Cabernet Sauvignon fermentations with octuplicate treatments on these coupons in the cap. And we cleaned them. We first did a cleaner-only trial, took our optimum cleaners, combined that with our optimal sanitizers, saw what worked the best, and then did our optimal cleaning sanitizing process as a third trial. Those that worked the best, and how did we test how they work? We did ATP swabbing and we did plate counts. Then we took the best from that and we went to the winery. We did, if you look at the bottom, it's a little bit busy, this slide. But basically what we found worked really well were our potassium caustic peracetic acid combination. There's other that worked equally well. We just thought this is the greenest option of those that worked well for us. We did both 5 and 10 minutes. We did a peracetic at 100 and 200 milligrams per liter. And we did the potassium caustic only at 1%. Right at the bottom, you'll see there's a potassium carbonate with PAA combination. That was just the standard process that was then applied in our winery. We did all of these in duplicate. And basically what I can say, if I compare our most stringent to our least stringent process, there wasn't really huge differences. What was really more impactful was not the cleaning and sanitization protocol that all worked pretty well, was more the areas that we swapped with ATP for ATP testing. So what you see in this graph is here at the bottom is really the sample location. Look at that. If you look BV21, BV22, you'll see the BV22, this is the gasket of the port door. BV23 was the interior surface of the tank wall above the port. Those areas weren't well cleaned because they were not getting any mechanical action. The spray ball wasn't hitting it well. So it doesn't matter if we saw 5 minutes, 10 minutes, it doesn't really matter. If it makes good contact, it will kill anything in 5 to 10 minutes. The problem is if it doesn't actually make decent contact, then it can't clean it properly and it can't sanitize it properly. And that's really the most important thing that we learned here. The other thing is if you look on the left, that large purple line there, hose. Our hose was cleaned with the same cleaning and sanitization. Everything was pumped through there. If we did a final rinse with that hose using water, we would have recontaminated our tank. Hoses are really, really hard to clean and sanitize. This is the standard rubber hoses. They crack very easily, microbes go into those cracks, and you just can't get it properly clean. We probably need to replace our hoses way more frequently than we actually do in wineries in general. Okay, so if you're not convinced yet, look at the right. That was the plating of the hose. And basically the gasket looked very similar to it. So our cleanest areas were the smooth surfaces. They were really clean. Bottom lines are really clean. Things that's usually not very clean, and we tested, drains, areas around the bung of barrels have high levels of microbial contamination. Pumps, hoses, thieves, winery process water. Check them all. Those are really areas that you can recontaminate something you just cleaned and sanitized. So basically what I'm saying, there's really no replacement for sometimes manual cleaning. If you clean a tank at the end, you need somebody to brush those gaskets, the ports, the valves. Those are the areas that's really difficult to clean, and manual cleaning of them needs to be included in your routine protocols and checklists. Very quick barrel management. Steam, there's been studies looking at Brettanomyces specifically and found that you need up to 12 minutes if you want no culturable cells at 9mm depth. We're also looking at this. So far, I haven't seen Brett moving in that deeply, but we're using new barrels, but many of these studies used older barrels that's been used many times. It takes an amount of time to get the inner wood surfaces to heat to 55 degrees, and only then does it start inactivating the yeast and bacteria. Ozone works really well. Like I said, you may have to make sure that you generate the ozone very close to your barrel. Chemical controls like ultrasonic, we don't have a lot of data on that. SO2, we're doing tests now in the winery using sulfur dioxide, both the gas and SO2 wicks. SO2 works really well, okay? It does what it's supposed to do. Our tests show that it kills everything that we've put in the barrel. Points to notice, cross-contamination of your barrels. Sampling and topping, you have to be really careful. Sanitize your wine thief during sampling in between every barrel. Critical that your topping wine is microbially stable. Use 70% ethanol sprays to clean the inner and outer side of your bung, both the barrel itself as well as the bung. So to summarize, basically there's no replacement for having good cleaning and sanitization protocols. Doesn't help you have a great protocol, but people don't follow it, skip steps. Don't believe your eyes, even if something is clean, following the cleaning protocol. Very important to also monitor that's actually working. ATP swabbing works really well. It's really quick. Come up with limits, what's acceptable values for your winery, and follow those steps. Anyway, so that is my main story. And be very careful, manual cleaning of gasket, hoses, drains, etc. Test them all, make sure that you've cleaned them properly. Thank you very much. Corey Marks did most of the work in this protocol, in this research. American Vineyard Foundation gave us, partly supported some of the research we did for this. And I also have to thank Ben Montpetit for giving me microbiology advice every time when I need that. And yes, I'm good. I'm hopefully didn't go over my time too much. Thank you very much.
Thank you, Anita.
Did you send me 20 messages that I didn't read?
I only sent you a few.
It's over. I apologize.
All right. Moving along to keep up with the time. Our next speaker is David Block. He's professor and the Sands Department Chair in the Department of Viticulture and Enology at UC Davis, and also has a joint appointment in the Department of Chemical Engineering at UC Davis. He's going to be talking about developing tools for predicting and optimizing phenolic extraction during red wine fermentations.
Great.
Okay, go ahead.
All right. Thank you. So what I'd like to do today is to tell you a little bit about some of the work that we've been doing for quite a while now on developing tools for predicting and optimizing phenolic extraction during red wine fermentations. I'm going to focus on some of the newer stuff. I'll mention some of our work that we did a while ago. All this work is in collaboration with Anita Oberholster's lab, as well as some of the early work that I'll talk about is in collaboration with Ian J. Gallo. The bulk of the work that I'm going to talk about today is done by a recent graduate student, Conrad Miller. So I'll give him credit up front and make sure I'll acknowledge him at the end as well. So what I'd like to do today is talk very briefly about the problem. Why are we concerned with developing tools for predicting and optimizing phenolic extraction? And then I'll talk about some of the tools. We'll talk a little bit about some of the data that we got in doing experiments in our winery, followed by the development of models for predicting phenolic extraction as a function of processing conditions. And then I'll show you a little bit about how you can use the model to give you an idea of how you can use the model to actually optimize your own processing in an industrial setting. All right, first of all, the problem. I think the problem is relatively straightforward. It's the phenolic extraction is complicated. If everybody could get exactly the phenolic profile that they wanted to in every red wine, we wouldn't really have to do anything for research. And yet it's complicated because you have multiple phases, you have liquid at the bottom, you have your cap at the top, you have the extraction of phenolics from the cap that gets mixed into the liquid during cap management events, pump overs or punch downs or things like that. You have mixing in the bottom, you don't have mixing at the top, you have heat transfer at the bottom, but not really appreciably at the top, and so forth. And all of this, all the phenomena that result in phenolic extraction are temperature and ethanol dependent. And so temperature and ethanol change spatially in the fermenter, and they change over time. And that's what makes it complicated. So going back probably about 9 years ago now, we started doing some experiments at our winery. This is with Anita and with collaborators at Ian J. Gallo to understand how fermentation parameters affected phenolic extraction. And we got some really good data on that. And I'm going to summarize a whole bunch of work in this one slide. These are some of the data that we got for phenolic extraction. I'm just going to focus on the lessons learned as opposed to the data. The first lesson we learned was that skin extraction happens early in fermentation, whether it's tannins or anthocyanins or any of those types of phenolics. If it comes from the skin, it happens early, and it's not very temperature dependent. The final level is not very temperature dependent. How quickly you get to that final level is somewhat more temperature dependent, but overall not very temperature dependent. The second lesson we learned is that seed extraction happens later than skin extraction, and it's definitely temperature dependent. The rate and the final level is very dependent on how hot you run your fermentation. The third lesson we learned was phenolic extraction is not a strong function of pump over volume or frequency. And in fact, no matter what volume or frequency we used, we saw almost exactly the same phenolic extraction at our scale, which in this case was 30-gallon scale in our TJ fermenters, our highly instrumented fermenters. In fact, when we went to the limit of that, we found that when we didn't pump them over at all, we still saw almost the same phenolic extraction as we did with frequent pump overs. And this is something that I have to say we didn't really understand at all. It was one of the reasons why we decided, okay, can we actually use the information we gained and some modeling techniques to actually predict what would happen, not only at the scale we were doing our experiments at, but at a commercial scale as well. And that's what led to the bulk of the work that I'll talk about today. All right, so let me go through how we developed our model and what kind of model we developed. So the model we developed was a mathematical model based on the physics of what's going on in a red wine fermenter. And the first model that we developed was to be able to predict temperature and ethanol concentration spatially in the fermenter and over time. And so there are various components of that. The first component of that model was a model for yeast growth. And that's the model that we have up here in the upper left-hand corner. And this is a model that we developed probably about 20 years ago or so, and published about 20 years ago or so. So this model predicts cell growth. Yeast cell growth is a function of nitrogen concentration. It also predicts ethanol formation and sugar utilization and so forth. The second part of this model, mathematical model, is basically the physics behind what's going on. So the first equation here is mass transfer, things like diffusion and so forth, of molecules away from the skins and seeds into the liquid. The second equation is the fluid flow for semi-compressible fluid. This means that this is the equation that's going to let the liquid move and the solids not move in the model. And the third equation is heat transfer. And that's heat transfer mostly taking away the heat from the liquid. Heat transfer from the solids in the cap really doesn't happen appreciably even when it's jacketed. And the final part of this model is to have some of the physical parameters that weren't in the literature, and that's things like the thermal conductivity of pumice over time. And Conrad Miller, the person who did this, because that wasn't available, went into the winery the first year he was working on this and just measured those parameters. All right, so altogether, this is the model. We put it together. We use something called finite elements to solve all those differential equations simultaneously. And we can predict what's going on three-dimensionally, for instance, with temperature as a function of space and time in a fermenter. So the first question is the model valid? And I'm not going to show you, we did a lot of work to prove that the model does really predict well. I'm only going to show you very quickly what we found in one case. And that's if we had a fermenter, and this is the cross-section, the temperature cross-section. You see the cap up here is hot, liquid's cooler. In this case, what we did was simulate a pump over, and then you can see the heat is well distributed, and then the cap heats up in between pump overs. So this is the model prediction. This is what we actually measured, and basically the same tank, or a very similar tank. And what you can see is the data looks very similar to what we predicted. And so that's just one of the methods we use to validate the model. We use a lot of other methods, and you're going to have to take my word for it, given the time that the model predicts really well, which we're very proud of because we didn't really fit the data at all. We just used the physics and parameters that we had measured previously. All right, so that's the model, predicts spatial distribution, temperature, and ethanol. What we really want to know is predict is phenolic extraction. And so for phenolic extraction, it's kind of complex in that we have the release of phenolics from seeds and skins, and then we have adsorption and desorption back onto the skins and seeds and any solids that are there. And then we have the reaction, things like degradation of anthocyanins, which happens when you get up to 35 degrees Celsius or so. And what we needed to do was all of these steps are a function of temperature and ethanol concentration. And so we need a model for that to combine with our original model that tells us how temperature and ethanol change over time and spatially in a fermenter. So that's our next model, and we want to put the two together. So this is the model that Conrad developed for phenolic extraction. And I'm not going to go into the details other than say there's a mass balance, there's release of molecules from both the skins and the seeds. And then there's a reaction step for, in this case, we just accounted for degradation of anthocyanins at higher temperature. And then there's a model for adsorption, desorption, all the parameters which Anita's lab measured in the lab during doing very detailed bench scale experiments. So this is the mathematical model. We went through and used our own data to calculate each of the parameters in this. And then what we did was we used that model, just that model alone, to look at small scale fermentations, which are more or less well mixed, and see how well they predicted. And what we can see is at different temperatures, here's at 20 degrees Celsius, we have the tannin here, and the blue is the anthocyanin. You can see at 35 degrees Celsius, in both of these cases, the model predicts really well for a well-mixed fermentation. But of course, the fermentations are not well-mixed as you get to larger and larger scales. And that's why the next step we want to do is put the two models together and see how well they predicted what happened when there's differences spatially and over time. So we have our model on the right, which is the for temperature, predicting temperature in ethanol. We add to that our model for phenolic extraction as a function of temperature in ethanol. And together that becomes our integrated model to predict phenolic extraction. So at this point, what we want to do is validate that model. Together, does it predict what happens experimentally? And so we did a lot of this, but I'm going to only show you again one data slide. And here we measured spatially tannins as a function of time in a red fermenter. And what you can see is the tannins are built up in the cap, and then they get distributed throughout during a pump over or punch down. And what you can see is in the blue and purple on the bottom, those are in the liquid. Those go up over time. The tannins in the cap also go up a little bit over time, but level out, and they get closer together as you continuously extract the seeds and the skins. So this is the experimental. This is the prediction from that combined model. And again, we didn't really fit the model to the data. We're just using the physics and this model for extraction. And so we're very happy to see that the model predicts very much what happens experimentally, which is a good thing. Okay, so given that we were able to validate the model, now the question is, if you have this model, what can you use it for? And first of all, I would say that one of the things that we used it for was to see how different your prediction would be if you account for all that spatial variability as opposed to not. And it turns out there's a lot of difference. So in order to get a good prediction, you do need to use this kind of combined model, because if you don't, you would get, for instance, a prediction of this blue curve on either side for anthocyanins on the left and tannins on the right. But as you go up in scale, you can see that the prediction, when we use this combined model, differs from a well-mixed situation. So when you go from small scale to large scale, you're going to get different results in extraction, and that's what this model shows. All right, so what's the practical use of this? You can what I like to think of this as an analogy is it's kind of like a GPS for achieving the wine phenolic profile that you're shooting for. You know, everybody has the GPS on their phone. It looks something like this. Think about the model as, you know, your location where you're starting from are the grape characteristics. So if you know what your grape characteristics are, your destination is that desired phenolic profile. The model allows you to predict, what should I do? What temperature should I use? How many pump overs should I use? How many punch downs or whatever it is to get, when should I press? To get exactly the outcome that you want. And maybe sometimes there's more than one route to get there, and the model could predict multiple routes, and you could use that to decide what your processing will be. Okay, so it could actually be a very useful tool to help you decide on processing. All right, so given that, what I'd like to do is just show you some case studies where we use the model to predict things, predict ways of processing to achieve different goals. All right, so these are just illustrations. So in case one, let's say our goal was to maximize skin tannin for whatever reason. It turns out that at a 2,000 gallon scale, what we predict is that actually the most skin tannin you're going to get is if you do pump overs once per day. And that's going to give you higher skin tannin than if you pump over 8 times a day or you don't pump over at all. So here's the yellow is the 8 times a day. The green is not pumping over at all. And that's because there's a trade-off between the more you pump over, the lower the temperature. But also the more you pump over, the more you lower the concentration of liquid in the cap, which speeds up release and overall extraction. And that's not necessarily an obvious result, something we get from the model. Second case study is, what if you wanted to minimize seed tannin? You're okay with, or the percent of tannin that comes from seeds. And so what we find from the model at either the 100 liter scale, 25 gallon scale, or the 2,000 gallon scale is with no pump overs, you see a higher percent seed tannin as a part of total tannin. And as you do the pump overs, you get reduced seed tannin. So in this case, reducing the seed tannin, increasing the pump overs will reduce the seed tannin. And this is mostly a function of a lower temperature because remember, seed tannin is very temperature dependent. And a third case study, this was really to understand that result that we got, that that no pump overs gave us just as good extraction as pumping over. We had anecdotally heard from people in industry that some of them told us, yeah, we've tried that by accident, and we also got good extraction with no pump overs. And we've been told by people within the same organization that they tried that by accident and found no extraction at all by not doing pump overs. And so what's going on with that? So we use the model to try and understand that. And what we figured out from that is it's not the volume that makes a difference. It's actually the difference in height, or the distance between the bottom of the tank and the bottom of the cap, that seems to make the most difference. So these data are for a 25 gallon short squat fermenter, that's the blue, and the tall skinny 25 gallon fermenter, that's the gray here. And then we have a 2,000 gallon fermenter that's very squat and wide, and then a tall and skinny 2,000 gallon fermenter. And what you can see is when the distance from the bottom of the tank to the bottom of the cap, like these two are the same, you get the same extraction. If you go to a short, wide, small fermenter, you get really good extraction without doing any pump overs. And if you have a 2,000 gallon fermenter that's tall and skinny and you don't pump over, you're not going to get any extraction at all. So we've used the model to explain these results that we couldn't explain just from the experiments. All right, so to summarize, we've developed a comprehensive physical model to predict temperature and phenolic gradients. We can use that in itself to examine fermentation design and processing, but we can also combine that with a phenolic model to try and to come up with a comprehensive model that then allows us, like that GPS, to use it to optimize future processing. To knowing what your grape characteristics are when you start, you can actually use a model like this to decide what you're going to do for processing to get the desired phenolic profile. So with that, I'd like to point out that I know this was fast, but I covered about 15 papers worth of material in about 20 minutes. You'll have access to the recording, so you can look up these papers if you want more information. You're also welcome to just email me. Here's my contact at the bottom. But I'd like to acknowledge the American Vineyard Foundation for funding a lot of this work, along with E&J Gallo and the Ernest & Gallo Endowed Chair for funding, and all of my collaborators, especially Conrad Miller, who did a lot of the work I talked about today, and Anita's lab, Anita Oberholster, for all of her collaboration over the years, in addition to the E&J Gallo team. So thank you.
All right. One more talk. Last but certainly not least is Ben Montpetit. He's an associate professor in the Department of Viticulture and Enology here at UC Davis. and he's going to talk about non-Saccharomyces yeasts, potential applications, and problems in wine.
All right. Thanks, Karen.
Thank you.
Can you hear me okay?
Yep.
Excellent. So being last, and I know with Zoom fatigue, it's a real thing, believe me, that I would, kind of present something a little bit shorter, lighter, and maybe kind of thought-provoking in a sense that I think this is an area of thinking about non-Saccharomyces yeast that really is going to see a lot of growth in the future. And because of the kind of potential that these organisms have to change wine fermentations and contribute to wine fermentations, as we know they already do. So I think historically, maybe most have thought about non-Saccharomyces yeast as being a source of spoilage. And of course, that is true in some instances, but what we know from a lot of work over the decades from many labs, including this work from Dave Mills' lab more recently, is that fermentations are this dynamic community of microbes that are working to metabolize the sugars and produce the sensory compounds that we think about. And this can be seen at the level of both bacteria as well as the yeast. And you see these over time going from the must through the end of fermentation. You see these changes in these populations. Some of these we know, obviously, and make sense to us. This is Enococcus, and it blooms close to the malolactic fermentation. And then this is Saccharomyces, which blooms during primary fermentation and then recedes over time. So we understand that the product produced is not, just from Saccharomyces, but from this community. And this has an impact on the outcome. One example of this is captured in this figure, which comes from a work looking at fermentations in New Zealand. And I picked it in large part because it's a nice schematic, I think, where they show the populations of the microorganisms in the juice and at the end of an uninoculated fermentation. And what we can see is that by site, you have this very large variability in the starting population of microbes and even in the ending population, where some fermentations go the way that you might expect in that they're dominated by Saccharomyces cerevisiae at the end of fermentation, where others at the end of fermentation, Saccharomyces is not the most dominant microbe. So knowing that there are these variabilities, why should we kind of think about this, and why should we think about what impact these non-Saccharomyces are having beyond spoilage per se? And that's because Saccharomyces itself pays attention to these other microbes in its environment and are interacting with these microbes through the metabolites that each yeast is producing. And while sometimes these can be negative and could lead to fermentation problems, they can either be additive or even synergistic, where the yeast together are having a better impact and outcome. And that this could be through sharing of macromolecules and one yeast, consuming compounds that are created by the other yeast. But there is competition as well, and that competition could be for nitrogen or other micronutrients, which I'll discuss in a little bit. So we refer to these as interspecies interactions. So these interspecies interactions are going on in the fermentation. And this has been realized for a number of decades, obviously. And even as back in 2012, when this review was written, it was noted that through yeast selection and using non-Saccharomyces yeast, you could manage to direct your fermentation to solve problems or change things in the way that you want. So whether that's responding to climate change or commercial trends or food safety issues, by combining the characteristics of these microbes and how the fermentation progresses, you could meet some of these challenges. And as I said, this is obviously happening between different yeasts, but I don't want to give you the sense that it only happens between yeast species. And it's well known and been published that these interactions also occur between bacteria and the yeast. So for instance, these three non-Saccharomyces yeasts, Pulcherima, Bacillus, and fermentans, have all been shown to increase levels of inhibitory compounds that impact the malolactic fermentation, while other organisms have been shown to actually stimulate the malolactic fermentation. So again, there is the potential here for site to influence the microbes that are present in your fermentation, and then for your fermentations then to take a course that is a reflection of the microbial community that's there. And of course, the idea would be is that if we understand these interactions and can then control these populations, that we could then influence the outcomes in a way that we wish. And people have suggested that by promoting certain non-Saccharomyces species, you could lower ethanol yields, you could affect manoprotein profiles, color stabilization, or in one instance, they even showed that you could replace the traditional malolactic fermentation by using 2 non-Saccharomyces yeast strains. So recognizing the potential here, the yeast companies have started to create products that can be used, and these are some of the species that are found in these dry active yeast products and some of the applications that are promoted by the companies based on some of the research indicated in the previous slides and others, of course. So these products are out there. You may be hearing about them. You may be talking to suppliers about them. And one kind of application that is being kind of discussed a lot right now is something called bioprotection. So bioprotection is as it sounds. It's the use of a biological agent to prevent the development of other spoilage microorganisms. And fairly simply, this is the idea that if you inoculate a must in this case with these non-Saccharomyces yeast that have this bioprotectant capability, what they're doing is basically filling that environmental niche and preventing other microbes from taking over. And this could have the advantage of limiting sulfite additions, for instance. Now, in some cases, the inoculated yeast can also have other benefits. That previous chart indicated, different uses, and in this case, there's information to suggest that some of these non-Saccharomyces yeast could lead to lower ethanol production, which could combat some of the effects of climate change long term. So what is the actual mechanism of action? How does bioprotection actually work? So the idea is that these yeasts that are inoculated either produce factors that kill the other yeast, so killer factor or killer proteins, or they produce other antimicrobial compounds that affect the growth of these other microbes. So Torulasporula, for instance, creates a compound called a hydroxytyrosol that is known to be active against bacteria and other yeast, for instance. The other way bioprotection is thought to work is it's simply that these yeast are competing for oxygen, lipids, and other nutrients in that environment. And by using those and competing for those, it limits spoilage. And this, of course, if it prevents spoilage, is a good thing. But we have to then consider that if this is how this process is working, that bioprotection is not specific then for the problematic organisms. By adding those non-Saccharomyces yeast, you are going to be changing the nutrient profile of the must, and that could ultimately impact the direction or the final product of the fermentation. So what this raises is the need for an understanding of the metabolism of these non-Saccharomyces yeast and how they use nutrients. But right now, I would say that this field is really largely unknown, and it may even be that the application of these non-Saccharomyces yeast requires the development of nutrients optimized for these yeast, which may not be the same as those nutrients we use now. So give one example of this was a recent study that looked at nitrogen utilization by a number of non-Saccharomyces yeast. And they did this at 2 different temperatures, and then they also looked at volatile compounds that were produced in sequential fermentations. This paper has a lot of data in it and a lot of interesting information for those of you who are interested in looking at it more. But to summarize in form of an example, what they found, for instance, is that some non-Saccharomyces yeast rapidly consumed some of the amino acid precursors, in this case, Bacillus rapidly consumed leucine, and that the lower level of leucine impacted ultimately isoamyl acetate levels in sequential fermentations. And there was other examples as well. And this simply comes about from the fact that when you inoculate with these non-Saccharomyces yeast, they're going to consume nitrogen and other micronutrients, and that those compounds are then not available subsequently for Saccharomyces cerevisiae. So again, this is an example where if you knew more about the nitrogen utilization information of these non-Saccharomyces yeast, you may be able to develop a product that either feeds these yeast specifically or replenishes these nutrients that are taken out of the must by the in these sequential inoculations. So if we know that these microbial fermentations are driven by a large community, of course the question becomes, how do we know what microbes are present, and how do we know if they're active and actually having an impact? And I'm just going to share with you a little bit of data from a recent or an ongoing study that we have looking at microbial populations from select sites. So what we've done in this is ask this question of what microbes are present on sites. These sites are defined by the same rootstock, the same clone of Pinot Noir, and are generally farmed in the same way. And what we've done is sampled the must, and I'm showing here just site-specific microbes. So there are many microbes found on this grape material that are exactly the same across the site. So there are many common grape-associated microorganisms. But across these different vineyard sites, which the abbreviations are defined here, you find unique microbial populations. So, some of these Russian River Valley sites or Santa Maria Valley sites are defined by unique microbial populations based on a DNA signature. So I forgot to mention this on the previous slide, is that what we're measuring here is the DNA signature. So this is the genetic material that's present, and its presence in whether the cells are alive or dead, active, inactive. So this is really telling us about a potential for these microorganisms to impact our fermentations. Now, what we've accompanied this with is information that comes from a related molecule called RNA. And the special thing about this molecule is that it's relatively unstable. So once cells become inactive or die, this molecule disappears and would no longer be present. So by measuring the presence of this molecule, we can assess whether the microbes that we're seeing are actually active or not. So the way we collected this information was that, again, from these same sites, we collected samples and measured these RNA signatures 16 hours after the yeast Saccharomyces cerevisiae was inoculated. So you could see something like these Arroyo Seco sites or these Anderson Valley sites that at this 16-hour time point post-inoculation, the vast majority of the sample that we're seeing, the signal that we're seeing, is coming from Saccharomyces cerevisiae. So this suggests Saccharomyces cerevisiae is the dominant microbe at this point, and it's the most active microbe. But this is not true of all sites. So these two Santa Maria Valley sites, for instance, are still dominated by non-Saccharomyces yeast at this point. So two hypotheses arise from this. Either there's something very specific about these musts that these non-Saccharomyces yeast very much like, and they're doing well, so they're competing with the Saccharomyces, or there's something about these musts that inhibit Saccharomyces, and therefore these microbes are able to persist up to this standpoint. And we don't know the answer to those two questions as of right now. But what I want to contrast for you is the information that we get from these two different approaches. So if we look at, for instance, Hanseníaspora uvarum, which if I go back one slide, this was a dominant organism, for instance, in these two Santa Maria Valley sites from these fermentations. If you look across all sites, using the DNA-based method where we look and ask what is present on the grapes, and this is in the must just prior to inoculation, we see that Hanseníaspora uvarum is there on all sites. And if you looked at this, you would say that if there are differences between these sites, it really can't be driven by uvarum. But when we look at the RNA-based information, what we see is that it's really only the Santa Maria Valley sites where these two microbes are present and really active. And if you looked at this information, you might suspect that non-Saccharomyces activities may be responsible for any unique characteristics coming from the Santa Maria Valley sites. So we're very much interested in pursuing this type of research where we're looking at these different pieces of information to get a better sense of what microbes are present and active in fermentations that could be driving site-specific characteristics. So, we're asking questions about what that starting microbial population is. And once we know about that population, we can then go farther into these issues that I've raised around interspecies interactions, and of course, what you can do to manage these fermentations to promote a given microbe or maybe inhibit the presence of that microbe over time. So with that, I'm going to finish. I just need to recognize the people that do the work. So Taylor and Rachel are two individuals who did a tremendous amount of work related to the microbiome sampling and RNA and DNA-based analysis of those samples, which I presented at the end there. And of course, recognizing the funders that make that work possible. With that, I will stop. And if there are questions, happy to take them, or I'll look in the Q&A and see if what is there.
All right, so if all of our speakers would like to show their video and unmute themselves, we can actually have sort of a Q&A or discussion with everyone. And we have Carrie on there as well.
Hello, thanks everyone. This turned out really good. Thanks so much.
So if anyone has any,
I was gonna say, it's like maybe they were expecting it wouldn't turn out good. Yeah.
Hey guys, this is almost like the Brady Bunch. Like I can look up,
it is. It
down,
does.
and so I'm only seeing. I will tell everyone that Caroline has put the link to Beth's survey in the Q&A. So in the Q&A, I believe you can copy and actually then paste that link. So if any of you guys want to take that survey. I also had gotten a question about about posting the slides, but actually the video of this, we're taking a video where it's being recorded. So we will post the video to Aggie Video, and I can send everyone who registered through A&R, I'll have people's emails and I can send them the link to that where that video resides on our Viticulture and Enology extension channel in Aggie Video. I'm not seeing any more questions though. Is there anything else that
you guys? Can I ask a question, Karen?
You absolutely can.
So I have a question for Dave Block, because we never see each other in person anymore and I don't get to hear him talk about research very much, but the last information that you shared there where it was tank size, et cetera, is that related to a surface area contact? Is that what you think is going on there, in the sense that when you have a short squat or a squat, very wide tank, that it's just that area of contact between the cap and the liquid? Or is there something more going on?
Yeah, I think it's partly the surface area of the bottom of the cap is large compared to the depth of the liquid, and that's allowing a faster extraction from the bottom of the cap.
Okay. So yeah, I was paying attention. I wasn't just having my video off.
There isn't a test, Ben.
That's true. That's true, right?
So if anybody that's still listening, if anybody has some concerns or issues in their area or things they would like us to research or to do extension programs on, you can share it in the Q&A in the chat, or you can email myself or Karen Block or anybody else you want to email. And we do really look at those suggestions when we put programs together. And it's always good to know what issues the industry are facing when we're thinking about grants, etc. as well.
Well, if there aren't any other questions, I would like to again thank all of our speakers. I'd also like to thank Carrie, for all your help. I know that we've been, like I said, we were back and forth a lot and putting this together over many months, hoping that we'd all get together in person back in April originally. And I really appreciate your sending it out and getting folks to listen here today. Appreciate that. I want to thank all of our sponsors, especially the American Vineyard Foundation, who gives us a grant each year to do these programs. And I'm really hoping that in the future we can do more of these in person. I really miss going out and being able to talk to people and you know, hear what their issues are in person. It is different, but we're trying.